Imagine a young man sitting alone in a massive, silent library. The only sound is the soft rustle of pages turning and the distant echo of footsteps on the marble floor. His name is Stanley Miller. He's a graduate student in chemistry at the University of Chicago, and right now, he feels both excited and completely overwhelmed. Spread out on the large wooden table in front of him are books, scientific papers, and his own notes filled with sketches and frantic writing. He isn't studying for an exam or working on a routine assignment. His mind is consumed by a question so vast, so fundamental, that it has haunted human beings for thousands of years. How did life on Earth begin? Think about that for a second. Look at your own hand. The skin, the bones, the blood flowing through the veins. It's all made of living cells, incredibly complex and perfectly organized. Now try to picture our planet, but 4 billion years ago. It was a terrifying and violent place. Volcanoes erupted constantly, spewing lava and ash into a sky without oxygen. The air was filled with poisonous gases. The oceans were hot and barren. There was no grass, no trees, no fish in the sea, no birds in the air. The world was utterly and completely lifeless. So how did we get from a dead rock in space to the beautiful, buzzing, overflowing with life planet we know today? How did non-living, simple chemicals transform into the first tiny living cell that would eventually become the ancestor of every plant, animal, and person that ever lived? This was the mystery that kept Stanley Miller up at night. It was a puzzle that felt both personal and universal. At the time, in the early 1950s, one of the most popular scientific ideas was called the primordial soup theory. It was a beautiful, almost poetic idea. Scientists like a brilliant man named Harold Urey, who was right there at the University of Chicago, suggested that the early oceans weren't just water. They were a rich, warm soup of simple chemicals, things like methane, ammonia, and hydrogen. And this soup was constantly being stirred by powerful forces. The intense heat from the young sun, the violent energy of lightning strikes, and the radioactive warmth from the planet itself. The theory was that over millions and millions of years, this energy acted like a cook, slowly coaxing those simple, lifeless ingredients to combine. They would form more complex molecules, then even more complex ones, and eventually somehow they would create the building blocks of life. These building blocks would then somehow assemble themselves into the first living organism. It was a wonderful story. But that's all it was, a story. A guess, a hypothesis. No one had any proof. No one had ever shown that you could actually take these simple chemicals, add energy, and get anything remotely close to the ingredients of life. It was like saying if you left a pile of bricks and wood in a field long enough, a house would eventually build itself. It sounded almost magical, and for a serious scientist, magic isn't enough. You need evidence. This is where Stanley, the young student, had his bold, almost crazy thought. He was sitting in that library reading Urey's papers, and the idea just clicked in his mind. What if I could test this? What if I could shrink the entire early Earth down to the size of a laboratory bench? What if I could put the primordial soup into a few glass flasks, replace ancient lightning with a simple electrical spark, and speed up 1 billion years of chemistry into just a couple of weeks? The idea was breathtaking in its ambition. He was just a student. Who was he to try and answer one of the universe's greatest secrets? People would surely think he was arrogant or foolish. They might laugh at him. You think you can create the beginnings of life in a bottle? They might say. You think you can solve in a few days what took nature an eternity? But Stanley wasn't arrogant. He was curious. He was driven by a deep human need to know. He believed in the power of experiment. Science for him wasn't just about thinking grand thoughts. It was about getting your hands dirty. It was about testing, failing, and trying again. He spent days, then weeks working on his idea. He sketched out designs for a strange-looking apparatus made of glass tubes and flasks. It would have a flask to act as the ocean, another to hold the atmosphere of gases, and a way to boil the water to create rain. Most importantly, it would have 2 metal electrodes to create a continuous miniature lightning storm, a tiny crackling spark of man-made energy. He knew his idea was a long shot, but he also knew he had one chance. He needed to convince the man whose work had inspired him in the first place. He needed to get the approval of the Nobel Prize-winning giant Professor Harold Urey. So one day, with his heart pounding in his chest, Stanley Miller gathered his notes and his carefully drawn diagrams. He walked down the university hallways, each step taking him closer to Professor Urey's office. He was about to propose an experiment that was either going to be a complete failure or one of the most important in human history. He took a deep breath, raised his hand, and knocked on the door. The sound of his own knuckles rapping on the heavy wooden door seemed incredibly loud to Stanley. It echoed in the quiet hallway like a gunshot. For a moment, there was only silence. His palms were a little sweaty. He clutched the folder in his hands a bit tighter. Inside were his notes, his sketches, the entire wild idea he was about to present to one of the most famous scientists in the world. "Come in," a voice called from the other side. Stanley turned the knob and stepped into the office. It was exactly what he imagined a Nobel Prize winner's office would look like, but also somehow more human. Books were everywhere, not just neatly on shelves, but in stacks on the floor piled on a corner of the large desk, their pages filled with notes sticking out like little paper flags. The air smelled of old paper, pipe tobacco, and coffee. And behind the desk sat Professor Harold Urey. Urey was a man who commanded respect without even trying. He had a sharp, intelligent face and eyes that seemed to notice everything. He wasn't scowling, but he looked serious, like a man whose mind was always busy with important, complicated thoughts. He looked up from the paper he was reading and waited. "Professor Urey," Stanley began, his voice feeling a little scratchy. "My name is Stanley Miller. I'm a graduate student in chemistry. I was at your lecture last week on the early Earth and the origins of life." Urey nodded slowly, gesturing for Stanley to take the seat across from him. "I see, and what did you think?" This was it. The moment to begin. Stanley sat down, leaning forward slightly, his enthusiasm starting to push his nervousness aside. "I thought it was fascinating," Stanley said, his words coming faster now. "The concept of the primordial soup, that the oceans were a broth of chemicals like ammonia, methane, and hydrogen, and that energy from lightning and the sun could have driven the reactions to create life's building blocks." Urey listened, his expression unchanged. He had heard this kind of praise before. He was waiting for the real reason for this visit. Stanley took a deep breath and plunged in. "Well, sir, it got me thinking. It's a brilliant theory, but it's still just a theory. What if we could test it? Not in a million years, but in a lab? What if we could simulate the conditions of the early Earth and see what happens?" He opened his folder and carefully slid his main diagram across the desk. It showed a drawing of a strange-looking glass apparatus. It had 2 glass flasks connected by a series of tubes forming a loop. "Imagine this," Stanley explained, his finger tracing the lines on the paper. "This first flask here is our ocean. We fill it with warm water. This larger one above it is our atmosphere. We pump in the gases you described, methane, ammonia, hydrogen. Then here," he pointed to a part of the drawing with 2 little lines drawn to represent metal, "we add 2 electrodes. We send a continuous electrical spark through the gases to act as our lightning storm." He looked up at Urey, trying to gauge his reaction. The professor's eyes were fixed on the diagram, his brow slightly furrowed in concentration. "The water heats up, creating vapor, just like the ancient ocean would have," Stanley continued, gaining confidence. "The vapor mixes with the gases in the atmosphere flask, and the spark acts on the mixture. Then a condenser cools it all down, so the water and whatever new chemicals form rain back down into the ocean flask, and the cycle repeats. We let it run for a week, maybe 2. We're essentially creating a tiny, speeding-up version of the early Earth's conditions." He finished and waited. The silence in the room felt heavy. The only sound was the faint ticking of a clock on the wall. Stanley's mind raced. Was it too simple? Was it a silly idea? Would Urey think he was just a naive student playing with things he didn't understand? Professor Urey leaned back in his chair, steepling his fingers. He looked from the diagram to Stanley's eager, anxious face. "Young man," Urey said, his voice calm and measured, "do you have any idea how complex life is? The molecules that make up a single cell are unbelievably intricate. What you are proposing is to create in a matter of days what nature may have taken millions of years to accomplish. The chances of you producing anything meaningful are extremely small." Stanley's heart sank. This was the rejection he had feared. He started to think about how to politely gather his papers and leave. But then Urey did something unexpected. A very slight, almost invisible smile touched the corners of his mouth. He leaned forward again, his eyes now sparkling with a hint of something, not quite belief, but a scientist's innate curiosity. "However," Urey continued, "that does not mean it is not worth trying. Science does not only advance by doing what is safe and likely to succeed. Sometimes the most important discoveries come from the long shots, from the experiments that everyone else thought were too difficult or too foolish to attempt. He tapped his finger on Stanley's drawing. This apparatus is elegantly simple. The logic is sound. If the theory is correct, this should at least produce something. Perhaps not amino acids, but perhaps some precursor, some hint of a more complex molecule. It would be a start. He looked directly at Stanley. I will give you 1 year. 1 year to build this, to run it, and to see what you can find. The odds are against you, Mr. Miller, but I believe in testing ideas. So let's test it. A wave of relief and pure excitement washed over Stanley. He couldn't quite believe it. He had permission. He had a chance. Thank you, Professor Yuri. Thank you so much. I won't let you down, Stanley said, practically jumping up from his chair. See that you don't, Yuri said, his tone serious again but not unkind. Now go. Get to work. And Mr. Miller, he added as Stanley reached for the door, keep detailed notes. Every single day. You never know what might become important. Stanley nodded, clutching his precious folder to his chest. He stepped out of the office and closed the door, leaning against the cool wall of the hallway for a second. He had done it. The impossible experiment was no longer just a dream in his head. It was now a real project with a deadline. He wasn't just a student with a wild idea anymore. He was a scientist with a mission, and he couldn't wait to get started. Stanley Miller walked out of Professor Yuri's office feeling like he was floating on air. He had done it. He had convinced a Nobel Prize winner to let him, a mere graduate student, attempt one of the most ambitious experiments imaginable. The excitement was a fizzy, bubbling feeling in his chest, like a soda bottle that had been shaken up. He wanted to tell everyone he passed in the hallway, but that excitement quickly turned into a different feeling, the heavy weight of reality. He had permission, but now he actually had to build this thing. He had to turn his simple pencil sketch into a working, real-world machine. His first stop was the university's glass-blowing shop. This was a magical place, hot and filled with the gentle hiss of gas flames. The air smelled of fire and hot glass. The glassblower, a practical man with strong, skilled hands, looked at Stanley's diagram with a skeptical eye. You want me to make this? The glassblower asked, pointing at the complex loops and connections.
Son, this looks like a fancy still for making something stronger than science. Stanley laughed a little nervously.
No, nothing like that. It's for a chemistry experiment. A very important one. The glassblower shrugged. He was used to strange requests from scientists. It's your time and money. This won't be easy. These joints have to be perfectly sealed. If there's even a tiny leak, your whole experiment is ruined. Stanley knew that. The gases he was using, methane, ammonia, hydrogen, were light and could escape through the smallest gap. If the seal wasn't perfect, his miniature atmosphere would just vanish into the air of the lab, and with it any chance of success. So, he waited. For days he paced outside the glass shop, peeking in to see the progress. He watched, fascinated, as the glassblower heated thick tubes of glass until they glowed a soft, bright orange. With a gentle breath and subtle twists of his tools, he shaped the molten glass, bending it into curves and carefully fusing separate pieces together. It was like watching an artist create a sculpture. Finally, it was ready. The glassblower presented Stanley with the finished apparatus. It was more beautiful and more fragile than he had imagined. It stood on a metal stand, a complex labyrinth of clear glass. The two flasks, the ocean at the bottom and the larger atmosphere flask at the top, were connected by a graceful loop of glass tubing. It looked like a piece of strange, modern art. Carrying it carefully as if it were made of eggshells, Stanley brought it to his lab bench. This was his space now, a small, cluttered table that would be the home for his miniature Earth. The next part was just as tricky, assembling the rest. He needed to make the lightning. He carefully installed 2 tungsten electrodes, small metal rods, into the atmosphere flask. These would be connected to a machine that could generate a continuous, powerful electrical spark. He attached a heating mantle under the ocean flask to keep the water warm and simulate the hot primordial seas. He connected a condenser, a cooled tube that would turn the steam back into liquid water, creating the rain. His lab bench was a mess of wires, glass, and clamps. It was a jumble of the ancient and the modern. He was using 20th century technology to recreate a world that was billions of years old. Other students and professors walking past his lab would often stop and stare. What was Miller building? It looked bizarre. Some would offer a helpful suggestion. Others would just shake their heads and smile, thinking it was another student's project that was bound to fail. Stanley ignored them. He was too focused. He would spend hours just checking the seals, making sure every connection was airtight. He remembered Professor Urey's warning. The chances are extremely small. He would look at his strange glass creation and feel a pang of doubt. Was this all just a waste of time? Was he just a fool playing with fancy glassware? But then he would remember the question, the big, beautiful question that had started it all. How did life begin? This glass contraption was his way of asking the universe that question directly. It was his way of listening for an answer. Finally, the day came. The apparatus was assembled. All the connections were tight. The electrical circuit was tested. It was time for the moment of truth. With a deep breath, Stanley began the final preparations. He carefully poured sterile, purified water into the lower flask, his ocean. He used a vacuum pump to suck all the modern air out of the entire system. The glass seemed to groan slightly under the pressure change. Then he opened the valves on 3 small tanks. He watched as the gases, methane, ammonia, and hydrogen, flowed into the empty glass world, creating its primitive atmosphere. He turned on the heater. The water in the ocean flask began to warm up. Slowly, tendrils of steam began to rise, traveling up the glass tube and mixing with the gases in the upper flask. The world was beginning to cycle. He stood before his creation. The warm water was simmering. The steam was rising. The gases were swirling. Everything was ready. All it needed now was the spark. His hand hovered over the switch for the electrodes. This was it. He was about to turn on a man-made lightning storm that would rage inside a glass jar. He was about to start the clock on his billion-year experiment. He took one last look at the clear, colorless liquid and the invisible gases. He said a silent, hopeful wish to no one in particular. Then he flipped the switch. A bright, sharp, continuous spark crackled to life between the 2 electrodes. It flashed with a blinding white light, illuminating the entire flask from within. The tiny, simple flame was burning. The experiment had begun. The sharp, loud crackle of the electrical spark was the only sound in the lab. Stanley Miller stood perfectly still for a long moment, just watching it. Inside the glass flask, the miniature lightning storm flashed and danced, a tiny, contained sun giving life to his small glass world. The water was heating up, sending steam into the gaseous atmosphere. The cycle had begun. For the first hour, it was mesmerizing. He couldn't look away. He checked the temperature of the water. He adjusted the flow of the condenser, making sure the rain was falling smoothly back into the ocean flask. He made notes in his lab book, his handwriting quick and excited. Experiment commenced at 10:17 a.m. Spark is strong and consistent. Cycle appears stable. But after a few hours, the excitement began to fade into something else. The boring, slow grind of waiting. Nothing was happening. Well, nothing he could see. The water in the lower flask was still clear. The gases were still invisible. The spark just kept on zapping, a relentless, repetitive flash. This was the part of science that nobody puts in the movies. It wasn't a sudden flash of genius. It was patience. It was routine. It was showing up every day, even when nothing seemed to change. Stanley fell into a routine. He would arrive at the lab early in the morning, often before anyone else. The first thing he would do was look at the apparatus. His heart would always jump for a second, hoping to see a change, a sign. But day after day, the water remained stubbornly clear. He would check all the connections for leaks, a daily ritual of tightening clamps and listening for the faint hiss of escaping gas. He would refill the water that had slowly evaporated over time. He would clean the glass where dust had settled. He became like a gardener tending to a very strange, very fragile plant that showed no signs of growth. The other people in the lab started to get used to his strange project. They would joke with him. "Any life in that soup yet, Miller?" one of them would ask with a friendly grin. "Not yet," Stanley would reply, trying to sound cheerful, "just giving it more time." "Maybe you should throw in some salt and pepper," another would call out. Stanley would laugh along, but inside the doubt was starting to creep in. What if Professor Urey was right and the chances were just too small? What if this was all a giant waste of a year? He was a graduate student. His entire future was on the line. Spending a whole year on a failed experiment wouldn't look good to anyone. He would sometimes catch Professor Yuri walking past the lab. Yuri would pause, look at the sparking apparatus for a moment, give a slow, unreadable nod, and then continue on his way. He never said anything. Was that a good sign? Or was he just checking in on his student's foolish project? Stanley couldn't tell. The days turned into a week. Then one morning, as he was doing his usual check, he noticed something. He leaned in closer, his nose almost touching the warm glass of the ocean flask. Was it his imagination? Or was the water not perfectly clear anymore? Did it have the faintest hint of a pinkish color? It was so subtle he thought it might just be a trick of the light, a reflection from the spark. He didn't write it down in his official logbook. He was too superstitious. What if he was wrong? Instead, he made a small, light pencil mark in the margin of his notebook. Just a tiny question mark. The next day he was sure of it. The pink tint was there. It was no longer his imagination. The clear, pure water was now definitely a very pale, rosy color. His heart started to beat a little faster. Something was happening inside the flask. The energy from the spark was breaking apart the simple molecules of methane and ammonia and forcing them to recombine into something new, something that had color. He continued his routine, but now with a new sense of anticipation. Every morning felt like a birthday, wondering what new thing he would see. The pale pink slowly deepened into a more noticeable rose color. Then, after a few more days, it began to turn a murky, brownish red. The liquid in his primordial ocean was now a dirty, ugly, beautiful brown. It looked like weak tea or old broth. It was definitely not clear anymore. The jokes from the other students stopped. Now, when they passed by, they would stop and stare. They would see the bubbling, muddy water and the constant flashing spark. "Whoa, Miller," one of them said, his voice serious now. "What is that?" "I don't know yet," Stanley would answer, a real smile on his face for the first time in weeks. "But it's something." The boring waiting was over. Now it was a different kind of waiting, an excited, almost nervous waiting. He had created a complex chemical soup. But was it just a bunch of random, messy chemicals? Or was it something more? Were the building blocks of life swimming around in that ugly brown water? He couldn't just look at it anymore. He had to know what was in it. The simple flame had done its job. It had cooked the soup. Now it was time to taste it. The next, most crucial phase was about to begin. The analysis. He had to find a way to look deep into that brown liquid and discover its secrets. The water in the flask was no longer water. It was a thick, murky broth, the color of strong tea mixed with dirt. After 2 weeks of constant cycling, of boiling, sparking, and raining, Stanley Miller's miniature world had transformed. The clear, simple beginning was now a complex brown mystery. The initial excitement of seeing the color change had settled into a new, more intense feeling, a deep, burning curiosity. It was like waiting to open a very important letter. He could see the envelope was there, but he didn't know what was written inside. What were the secrets of his homemade primordial soup? He knew he couldn't just guess. He needed proof, solid scientific evidence, and for that, he needed to use a technique that was, at the time, one of the best ways to separate and identify unknown chemicals: paper chromatography. It sounds complicated, but the idea is beautifully simple. Imagine you have a drop of black ink. Is it just one color? Or is it a mixture of blue, red, and yellow all blended together? To find out, you could put a drop of the ink on a paper towel and then touch the edge of the paper to water. The water would travel up the paper, carrying the ink with it, but the different colored dyes in the ink would travel at different speeds, separating into distinct bands of color. That's chromatography in a nutshell. For Stanley, the ink was his brown chemical soup, and he was hoping to find specific colors in that soup, the building blocks of life known as amino acids. The first step was to stop the experiment. One morning, after carefully noting the final conditions in his logbook, he took a deep breath and flipped the switch. The loud, constant zit-zit of the spark suddenly stopped. The lab fell into an eerie silence it hadn't known for 2 weeks. The only sound was the gentle simmer of the hot water. The man-made lightning storm was over. The cooking was done. Now he had to work carefully. He couldn't contaminate his sample. If he got even a tiny bit of his own skin or hair in it, it could ruin everything, fooling him into thinking he had found something that was actually just from himself. He used sterile glass pipettes, his hands steady but his heart racing, to draw out a small amount of the brown liquid from the ocean flask. It was a delicate operation, like a surgeon making the first cut. He prepared his samples, mixing them with specific chemicals to make any potential amino acids easier to detect. Then he took a special large sheet of filter paper. With a pencil, he lightly drew a line near the bottom. Along this line, he carefully placed tiny, concentrated drops of his precious broth. The next part was like a magic trick. He placed the paper into a sealed glass tank that had a small pool of a special solvent at the bottom. This solvent would be the water that traveled up the paper towel. He closed the lid and waited. He couldn't do anything now but watch. Slowly, almost invisibly, the solvent began to creep up the dry filter paper. As it passed through the spots where he had placed his brown drops, it began to pick up the chemicals and carry them upwards. The different chemicals in the mixture, depending on their size and structure, began to race up the paper at different speeds. After several hours, he carefully removed the paper. To the naked eye, it didn't look like much. It was just a wet piece of paper with some smudgy, brownish streaks on it. The real magic would happen next. He needed a way to see the invisible. He used a chemical spray that would react with amino acids. He sprayed the paper evenly and then gently heated it. He held his breath. For a moment, nothing happened. The paper was just wet. Then slowly, like a photograph developing in a darkroom, faint spots began to appear. They started as vague purplish smudges, then grew darker and more distinct. His eyes widened. He leaned in closer, his nose almost touching the paper. There they were. Not just one, but several. A series of dark purple spots, each at a different height on the paper. Each spot represented a different chemical that had been in his soup. His hands trembled slightly. This was the moment of truth. He had a map, but he needed the key. He ran what is called a control experiment. On the same kind of paper, he put drops of known pure amino acids, glycine, alanine, and others. He let the solvent carry them up, sprayed them with the same chemical, and heated them. These known amino acids created spots at specific predictable locations on the paper. Now came the comparison. He held up the two papers side by side against a light. One was from his experiment with its mysterious purple spots. The other was his control, with spots whose identities he knew for sure. He looked from one to the other. His heart was pounding in his ears. He lined up the spots there, the spot from his flask. It lined up perfectly with the spot for the amino acid glycine. He moved his eyes. Another spot from his experiment matched exactly with the spot for alpha-alanine, and another for beta-alanine. It wasn't just a messy soup of random chemicals. It was a precise, organized collection of molecules. The simple flame, acting on simple gases, had created the fundamental building blocks of proteins, the very machinery of life. He stood there in the quiet lab, staring at the two pieces of paper. The proof was right in front of him in the form of simple purple circles. He had done it. He had taken the raw materials of the early Earth and, in just 2 weeks, created the ingredients for life. A huge, overwhelming wave of emotion hit him. It was a mix of triumph, disbelief, and a profound sense of wonder. He wasn't just looking at chemicals on a piece of paper. He was looking at a powerful message from the past, a message that said, "This is how it could have started. It's not magic. It's chemistry." He carefully, reverently placed the two pieces of paper on his lab bench. He had to tell Professor Urey. The long shot, the foolish student's project, had actually worked. Stanley Miller stood in the lab, staring at the two pieces of filter paper laid side by side on the bench. The purple spots were undeniable. They matched. He had created amino acids, the building blocks of life, in a glass jar. The feeling was surreal, like he had just discovered a new law of nature in this small, cluttered room. His first instinct was to run, right that second, to Professor Urey's office. But he stopped himself. A good scientist is careful. A good scientist checks his work. What if he had made a mistake? What if it was a coincidence or worse, contamination? He couldn't go to a Nobel Prize winner with results that might be wrong. For the rest of the day and all of the next, he became a detective investigating his own discovery. He repeated the chromatography process. He cleaned his glassware again and again, even more meticulously than before. He ran new samples from his brown broth, using different solvents to double-check the results. Every single time, the answer was the same. The purple spots appeared in the exact same places. Glycine, alanine. They were really there. The evidence was solid. Finally, his confidence outweighed his fear. He gathered his lab notebook, the original chromatogram papers, and his repeated results. He arranged them neatly in a folder. His hands were steady now, not from calm but from a focused certainty. He walked the now familiar route to Professor Yuri's office. This time the walk felt different. The first time he was a hopeful student with a wild idea. Now he was a researcher with proof. He knocked on the door, the sound firm and clear. "Come in, Miller," Yuri's voice called out. It seemed he was almost expecting him. Stanley opened the door and found Yuri at his desk, surrounded by his usual fortress of books and papers. Yuri looked up, his sharp eyes taking in Stanley's appearance, the folder, the serious but excited look on his face. "Professor," Stanley began, his voice clear and strong. "The experiment is complete. I've analyzed the results." Yuri gestured to the chair. "Sit, show me." Stanley sat down and opened the folder. He didn't launch into a big speech. He simply laid the evidence on the desk. He placed the two key pieces of paper side by side, the one with the spots from his experiment, and the one with the spots from the known amino acids. "Sir, as you can see," Stanley said, pointing with his finger, "this spot here from the flask liquid aligns perfectly with the reference for glycine, and this one here is a perfect match for alpha-alanine. I've repeated the analysis 3 times. The results are consistent." He then slid his detailed lab notebook across the desk, open to the pages where he had recorded the color change day by day, and his meticulous notes on the chromatography process. Professor Yuri leaned forward. The casual, almost bored expression he often wore vanished completely. It was replaced by an intense, piercing focus. He didn't say a word. He picked up the 2 chromatogram papers and held them close to his face, his eyes scanning back and forth between the spots. He compared them to the notes in the lab book. He was checking the data line by line, just as Stanley had. The silence in the room was heavy. It felt like it lasted for an hour, though it was only a minute or 2. Stanley held his breath. This was the moment of judgment. Would Yuri find a flaw? Would he dismiss it? Yuri slowly lowered the papers. He looked up at Stanley, and his face underwent another transformation. The stern, unreadable mask was gone. In its place was a look of pure, unadulterated astonishment. His eyes were wide. He leaned back in his wooden chair, which creaked under his weight. He was silent for a few more seconds, just staring at the results, then at Stanley. Then he spoke, his voice quieter than Stanley had ever heard it, filled with something like reverence. "My God," Yuri whispered. "You did it. You've actually done it." He looked from the papers to the young student sitting across from him. "You've proven that the building blocks of life can be made from the basic ingredients of the early Earth. This is not just a student's project anymore, Miller. This is a fundamental discovery." The weight of those words sank into Stanley. Hearing it from Yuri, a man who had won the highest prize in science, made it real in a way that his own excitement couldn't. Yuri was now energized, his mind racing ahead. "We must publish this immediately. The world needs to see this." He picked up the chromatogram again, shaking his head in wonder. "The implications. This changes everything. It provides a plausible, testable path from chemistry to biology." He looked at Stanley, a new respect in his eyes. "You have done in 2 weeks what people have been speculating about for centuries. This is outstanding work, truly outstanding." For Stanley, the professor's reaction was the final, confirming piece of the puzzle. The validation from this brilliant, hard-to-impress man was more rewarding than any prize. The long nights, the doubt, the jokes from other students, it all melted away in that moment. He wasn't just a student anymore. Under Yuri's proud gaze, he felt like a fellow scientist. They had started with a simple sketch on a piece of paper, and together they had opened a window into the very origins of life itself. The journey was far from over, but the first and most important step had been proven. It was possible. The quiet, focused world of Stanley Miller's lab was about to be turned upside down. Professor Yuri, a giant in the scientific community, had given his stamp of approval. The next step was to share their discovery with the world. This meant writing a paper, a formal report of their experiment and findings, to be published in a major scientific journal. Stanley sat down with Yuri to write it. There was a big question from the start. Whose names would go on the paper? In science, the order of names is very important. The first name is usually the person who did most of the hands-on work. the main driver of the experiment. The last name is often the senior scientist. The professor who guided the project and provided the lab, Yuri, in a move that showed his immense respect for his student, insisted, "Miller, your name will be first. This is your work, your idea, your hands that built it, your patience that saw it through. I was merely your guide." And so the paper was written under the names Stanley L. Miller and Harold C. Urey. It was a short, powerful report, only a few pages long. It didn't make grand claims about creating life. It was careful, precise, and humble. It simply described the apparatus, the conditions, and the undeniable result: the formation of amino acids from simple inorganic gases. They sent it to the most prestigious science journal in the world, Science. Then, more waiting began. But this waiting was different. It was filled with a nervous, electric anticipation. Stanley went back to his daily routine, but now every ring of the phone or piece of mail made his heart jump. Would the journal accept it? Would other scientists rip it apart? Weeks later, the response came. The paper was accepted. It was scheduled for publication in the May 15, 1953 issue of Science. The day the journal finally arrived on campus was a day Stanley would never forget. He held the physical magazine in his hands. There it was in black and white for anyone in the world to read. A production of amino acids under possible primitive Earth conditions, by Stanley L. Miller. He showed it to his family. He showed it to his friends. It was real. He was a published scientist, and not just with any paper, but with one that felt truly important. At first, the reaction was a slow rumble. Scientists at the University of Chicago read it, then scientists across the country, then the world. The scientific community was stunned. It was like a thunderclap. For the first time, someone had provided hard, tangible, repeatable evidence for how life could have started. It wasn't just philosophy or guesswork anymore. You could go into any lab on Earth, build Miller's apparatus, and see the same results. The primordial soup theory was no longer just a theory. It now had a solid foundation. Newspapers and magazines picked up the story. Reporters, who usually wrote about politics or sports, suddenly wanted to talk about methane, ammonia, and the origins of life. Stanley Miller, the quiet graduate student, found himself being interviewed. His picture was in the papers. The headlines were dramatic. "Scientist creates building blocks of life in test tube. An experiment may solve riddle of life's beginning." People were fascinated. For the public, it was both exciting and a little frightening. The idea that the secret of life could be so simple was mind-boggling. It made people look at the world differently. The spark of life seemed less like a magical mystery and more like a natural process, a beautiful and powerful kind of chemistry. Of course, not everyone was happy. Some religious leaders were concerned. They felt that science was stepping into territory that belonged to God. They worried that an experiment like this tried to remove the divine from the creation of life. This created a new public conversation, one that continues to this day, about science, faith, and how we understand our own existence. Back in the lab, Stanley handled his new fame with a quiet humility. He wasn't a showman. He was a scientist. While the world was buzzing with excitement, he was already thinking about the next questions. Was this the only way it could have happened? What about other energy sources like volcanic heat or ultraviolet light? What other molecules could be formed? He had opened a door, and now he could see a whole new hallway of mysteries waiting to be explored. The simple flame in his glass apparatus hadn't just created amino acids. It had ignited a new field of scientific research and had given humanity a new, profound story about its own possible beginnings. The young student was now a pioneer. For a long time, Stanley Miller lived in the bright light of his discovery. He became a respected professor himself, teaching and continuing his research at the University of California. Wherever he went in the world of science, he was known as the man who had recreated the origins of life in a flask. His experiment was in every textbook. It was a classic, a perfect story of scientific triumph. But science never stands still. It is a living, breathing process of asking new questions, finding new evidence, and sometimes challenging old ideas. The very thing that made Miller's experiment so powerful, that it was a testable, specific model of the early Earth, would eventually become its greatest vulnerability. The problem started with rocks. Very, very old rocks. Geologists, scientists who study the Earth, began to find and analyze rocks that were billions of years old. These rocks were like time capsules. By studying their chemistry, geologists could make educated guesses about what the Earth's atmosphere was like when those rocks were formed, and their findings started to paint a different picture from the one Harold Urey had proposed. Urey and Miller's model used a reducing atmosphere, one rich in hydrogen, with gases like methane and ammonia that are ready to give away electrons. This kind of atmosphere is perfect for building complex molecules, which is why it worked so well in the experiment. But the ancient rocks began to tell a different story. The evidence started to suggest that the early Earth's atmosphere might not have been a strong reducing one. Instead, it might have been more neutral, made up mostly of carbon dioxide and nitrogen, the same gases we breathe out and that make up most of our air today. These gases are much more stable, much less reactive. They don't like to form the kind of complex chemical bonds that create amino acids. When other scientists tried to run the Miller-Urey experiment with this new mixture of carbon dioxide and nitrogen, the results were disappointing. The spark would flash, the water would cycle, but the water stayed clear. No beautiful, complex brown broth formed. Only tiny, insignificant amounts of organic molecules were created. This was a major problem. It was like finding out that the key ingredient in a famous recipe never actually existed in the first place. The scientific community, which had once celebrated Miller, began to whisper. At conferences, other researchers would present papers questioning the relevance of his 1953 experiment. The whispers weren't mean-spirited, but they were persistent. Miller's work was elegant, one might say, but was it relevant? Did it really tell us about our Earth or just about a possible Earth? The early atmosphere probably wasn't like that, another would add. It's a beautiful demonstration of chemistry, but perhaps not a true history. For Stanley Miller, now an older man, this was difficult to hear. He had spent his life building on this discovery. To have the very foundation of his life's work questioned was not just a professional challenge. It was personal. He had seen the amino acids with his own eyes. He knew the power of that simple spark. Could it really be that his experiment, so famous and so clear, was just a beautiful, lucky mistake? A demonstration of a process that never actually happened on Earth? He defended his work. He argued that there could have been local environments on the early Earth, near volcanoes or in specific regions, that had the right reducing conditions. Perhaps the building blocks of life formed in these special pockets, not everywhere at once, but the evidence from the rocks was strong. The wind had gone out of the sails of the primordial soup theory. The Miller-Urey experiment was still taught in schools, but often with a caveat. This was a groundbreaking experiment that showed it was possible, but we now believe the early Earth's conditions were probably different. The bright, shining story of 1953 now had a long, slow shadow of doubt cast over it. Stanley Miller continued his work, but the world had largely moved on, searching for new answers to the old eternal question. His famous apparatus was packed away, a museum piece from a forgotten battle. It seemed the final chapter on his great discovery had been written, and it was a chapter of graceful but definite decline. The simple flame, it appeared, might have only lit up a dead end. As the years passed, Stanley Miller grew older. The fierce debates about the early Earth's atmosphere slowly quieted down, not because they were solved, but because science had found new puzzles to capture its attention. His famous experiment, once a revolution, was now treated with polite respect, like a retired champion who people remember fondly, but don't talk about much anymore. He moved his lab from the University of California to a new location. In the process of packing, like anyone who has accumulated a lifetime of work, he had to decide what to keep and what to throw away. There were boxes filled with old papers, notebooks with faded handwriting, and outdated equipment. In a corner of his lab, tucked away, was the original apparatus from 1953. It hadn't been used in decades. The glass was dusty. It looked fragile and old, a relic from a different time. A less sentimental person might have seen it as junk, a complicated piece of glassware taking up space. But Stanley couldn't bring himself to discard it. It was more than just glass and wires to him. That apparatus was the physical proof of his boldest idea, the vessel that had held his miniature world. It represented the most exciting and defining period of his life. So he carefully packed it into a sturdy box. He didn't just throw it in. He wrapped it and cushioned it, making sure it would survive the move. Along with the apparatus, he packed several cardboard boxes. These were his archives. They were filled with vials and notebooks from not just the famous 1953 experiment, but from many other experiments he had conducted throughout his career. To anyone else, they were just boxes of old, probably useless samples. But to Stanley, they were his history. They were the raw data of his life's work. The boxes were moved to his new lab space, and then, as these things often happen, they were largely forgotten. They were placed on a high shelf or in a storage closet, out of sight and out of mind. The world outside had moved on, and these boxes sat in silence, gathering a fine layer of dust. Stanley Miller continued to teach and do research, but he never again captured the world's imagination like he had in 1953. He watched as younger scientists proposed new theories for the origin of life. Some suggested it began in the intense heat and pressure of deep-sea volcanic vents. Others looked to clay minerals or even to outer space. The primordial soup was no longer the star of the show. He must have felt a certain sadness. It's a human feeling to see your greatest achievement, something that once made you famous, slowly fade from the spotlight. He still believed in his work. He knew what he had seen. But the evidence against the specific conditions of his experiment was strong, and he was a scientist enough to respect that evidence. Then, in 2007, Stanley Miller passed away. The news spread through the scientific community. Obituaries were written, celebrating the man who had once dared to ask the biggest question in a lab bottle. They spoke of his classic 1953 experiment with great respect, remembering the shockwave it had sent through the world. It was a chapter finally closing. After his death, his colleagues and former students began the sad but necessary task of cleaning out his lab. It was a time for remembering and for sorting through the physical remnants of a long career. It was during this process that someone, perhaps a former student named Jeffrey Beta who had worked closely with Miller, noticed the boxes. They were tucked away on a shelf, labeled in Miller's own handwriting. Curious, they opened one. Inside, neatly organized, were rows of small glass vials. Each vial was carefully sealed and had a faded label with dates and codes. These were the actual samples from Miller's experiments, preserved for over 50 years. Most people would have seen this as a job for the trash bin. They were just old chemicals. But Beta, a scientist himself, looked at them differently. He saw a treasure. He knew that the technology for analyzing chemical samples had advanced in ways Miller could never have imagined in the 1950s. The tools they had now were millions of times more sensitive. He held one of the vials up to the light. The liquid inside was still there, a silent witness to a long-ago spark. A thought both exciting and solemn crossed his mind. What if they were wrong to dismiss Miller's work so completely? What if the real story, the true power of that simple flame, was still hidden inside these vials, waiting for the right tools to reveal it? The answers weren't in a new theory or a new experiment. The answers were right here, in this old box, in the careful handwriting of a young student who had once changed the world. The story wasn't over after all. Jeffrey Bata, a former student of Stanley Miller, stood in the quiet lab, holding one of the dusty vials. He felt the weight of history in his hand. This small glass container held a piece of his mentor's legacy, a legacy that the world thought it had fully understood and then largely set aside. He made a decision. He would not let these archives be thrown away. He would give them one last look, using the powerful tools of the 21st century. He gathered a small team of young, curious scientists. They were fascinated by the story of Miller's experiment. To them, it was a legend from the history books. The idea of testing the actual samples was like being given a key to a time capsule. They carefully selected a few vials. One was from the famous 1953 experiment. But Bata knew Miller had done other, less famous versions of his experiment. He found vials from an experiment where Miller had added a twist. He injected steam into the spark, simulating a volcanic eruption. These samples had never been properly analyzed with modern equipment. Back in the 1950s, the methods just weren't sensitive enough. The team got to work. They used a machine called a high-performance liquid chromatograph, coupled to a mass spectrometer. In simple terms, this machine is a super detective for molecules. It can take a tiny drop of liquid, separate every single chemical inside it, and then identify each one with incredible precision. It can find things that would have been completely invisible to Miller and his paper chromatography. The process was slow and meticulous. They handled the old vials with the care of archaeologists handling ancient artifacts. A young researcher carefully prepared a tiny sample, just a few drops, and injected it into the machine. Everyone gathered around the computer screen. A graph began to appear. It was a landscape of peaks and valleys, each peak representing a different molecule that the machine had found. At first, they saw what they expected. The peaks for glycine and alanine were there, clear and strong, confirming Miller's original work. But then the machine kept going. More peaks began to appear, dozens of them. Then more than 100. A silence fell over the room, broken only by the hum of the machine. The scientists stared at the screen, their eyes wide with disbelief. The graph was a crowded, messy, beautiful skyline of molecules. Miller's simple broth was far more complex than anyone had ever dreamed. The young researchers, with their modern tools, were discovering that Stanley Miller's experiment had been even more successful than he ever knew. His simple flame had created a stunningly diverse cocktail of organic compounds. They identified more than 20 different amino acids. But it didn't stop there. They also found the building blocks for other essential molecules of life, lipids for cell membranes, and most importantly, the precursors to nucleotides, the pieces that make up DNA and RNA. The volcanic simulation vials showed even more dramatic results. The combination of intense heat and electrical spark had created an even richer, more diverse array of molecules. The implications were staggering. The scientific community had dismissed Miller's work because the specific atmosphere he used might have been wrong. But this new discovery showed that the principle was powerfully, undeniably right.
Energy acting on simple common chemicals could produce an astonishing treasure trove of life's ingredients under a variety of conditions.
The path from chemistry to biology was not a narrow, fragile one, but a robust and fertile highway. When Bada and his team published their new findings, it sent a new, quieter, but more profound shockwave through the world of science. It wasn't a rejection of Miller's work. It was a glorious vindication. The story was picked up by news outlets around the world. The headlines this time were different. They spoke of a lost experiment and a vindication. The narrative changed. Stanley Miller was no longer just the author of a beautiful but possibly irrelevant experiment. He was a visionary whose work had been decades ahead of its time. The true depth of his discovery had been hidden, waiting for technology to catch up. The simple flame in the glass apparatus had never gone out. It had continued to burn quietly, in the sealed vials and in the determined mind of a scientist who had saved them. Stanley Miller had passed away, but his curiosity had lived on in his students and in the physical evidence he so carefully preserved. His journey taught the world a final, beautiful lesson about science. It's not about being right on the first try. It's about asking a brave question, doing the work with your own hands, and leaving a trail for others to follow. The search for how life began is not over. But because of a persistent student and his boxes of old samples, we now know the universe is even more creative and more ready for life than we ever imagined. The spark he ignited continues to light the way.